Yeah, good afternoon and a warm welcome to this episode on the latest research on COVID-19. My name is Matthias Schneider. I'm a professor for medical and biological physics at the University of Dortmund in Germany. I'm also one of the members and initiators of the No COVID team, the COVID Action Group, and the World Health Network. Today, we want to talk about the latest research on long COVID or the post-COVID syndrome. But before we start, our guest speaker, which is Professor Jochen Guck from Erlangen, I want to give you a brief introduction in what the problem is actually all about. So one of the main topics around epidemics right now is vaccination, of course. And many people had their hopes up that this will be the nail in the coffin of COVID-19. But to rely 100% on vaccination is not a good idea at all. And let me tell you why. One reason is that although protection against hospitalization and severe disease may be very efficient, and it looks like from the latest studies of the CDC, around 95%. Vaccinated people, when affected, though, can still transmit the virus. This comes from the same data reported recently, only this week from the CDC. So, and the rate of transmission is even comparable to those people who have not been vaccinated. And this, of course, has some serious implications. Some people cannot be vaccinated, so they are unprotected, like kids under the age of 12 years, which in Germany are roughly 10 million. Some people are only poorly protected by vaccines, such as people that are immunocompromised, cancer patients, elderly, numbers given also in the same report in the order of 60, 70, 80% protection instead of 95. So I think our social contract demands us to treat these people with respect, on brief, meaning that we have to apply the golden rule. We should treat them the way we want to be treated in theirs, if we would be in their shoes. There's another reason, though, why we cannot simply rely on vaccines, and this is what we're going to talk about today. And this is the so-called long COVID or post-COVID syndrome. And I don't want to dissect it in details. I just want to briefly tell you it's something that can show in many shape and forms. Very typical is fatigue. Very typical is also a shortness of breath, cognitive disturbances, like people have sleep problems, people have problems with smell, the sense of smell, or headaches. But there's also problems reported with the heart, with the circulatory system, and the kidneys. But what does long mean in long COVID? And what you see here, this is a graph from the Office of National Statistics. And you see in the upper graph, the blue one is on the y-axis is the percentage of patients that have been positive with COVID-19 who report after, what the dotted line is, after roughly 12 weeks, still symptoms. And the green curve below is the control group. This means people who have been either COVID negative or have never been diagnosed. So you see that about roughly, this is 14% versus 2%, rather a factor of 7 more likely that you have long-term effects, long-term symptoms, the way I was mentioning them, such as fatigue, shortness of breath, and so on. And many of them suffer over several months, so even after 4, 5 months, 6 months. And these people do not just report some shortness of breath or some fatigue issues. Some really, like 2 to 3%, report after several months severe limitations in their daily activity. And in severe limitations means that certainly that they cannot go to work and cannot follow their work and their regular activities. Okay, this is all I had to say I wanted to say as an introduction. But now let me briefly introduce our speaker. It's my great pleasure to have Professor Jochen Guck here. He's the director of the Max Planck Institute of Light in Erlangen, where he is head of the biological and optomechanics section. Jochen and myself actually go way back, all the way to high school, and we kept in touch since. I admire his work. He did some impressive work in which he looks at biological material from a physical angle. He's one of the pioneers in what I call sometimes mechanical diagnostics of cells. and build the tools and theory around this. Jochen, it's my great pleasure to have you, and tell us a little bit about your work on long COVID. Let me actually get out of here, and I think you should be free to share your screen now. I think you have brought a few slides for us. Is that correct?
I did indeed.
Excellent. This looks good. Okay, Jochen, stage is yours. Thank you very much for being here.
So thanks, Matthias, for the introduction and for the invitation to present my work here in general. As you said already, I'm a director at the Max Planck Institute for the Science of Light, in German die Physik des Lichts. I'm also involved in setting up a new Max Planck Zentrum für Physik und Medizin. So this is getting a whole lot closer to today's topic than just using light. And the title of my presentation is Feeling for COVID-19, which might sound a little bit strange. What I mean by feeling for, you kind of gave it away already that we're interested in mechanics and semi-mechanical properties. So I'm a physicist by training, but I've been working for the last more than 2 decades now already on biological samples, in particular studying the physical properties of cells and trying to figure out how important these physical properties are for the normal biological functioning of cells, and also how important they are in order to figure out what is going on in disease. So we want to translate our insight into clinical applications ultimately. So why do we think that mechanical measurements are a good idea? I want to motivate this by taking you to a supermarket, and we are tasked with buying tomatoes. So what you see on the screen here is obviously a tomato, right? It's round and red and has this green funny stuff on the top, so it's easy enough to spot the tomato. So I guess you can easily maneuver yourself to the stand with the tomatoes in the supermarket. What do you do next? What you typically do, I guess, is you grab one of those tomatoes and you squeeze it, right? You want to know whether it's firm, whether it's ripe, whether it's too ripe, whether it's too soft. So you are very much in tune with the idea that a mechanical measurement can give you important information about the object that you're dealing with, right? That's just common sense and daily experience. Also, if you think about it in the medical world, doctors often use a manual palpation to figure out what is going on with the patient. A female that has a breast cancer, I mean, a tumor can be detected by manual palpation. Also intrasurgery, doctors sometimes feel for the texture, the stiffness of the tissue in order to figure out what they should still remove or not in the context of cancer surgery. So also in the medical field, I mean, a manual test actually can help hold important information. Now, we want to transfer the same idea down to the single cell level. So we want to take single cells, squeeze them, see how deformable they are, and then try to infer processes that are going on in the cell that are normal physiological processes, but maybe also pathological processes by this mechanical testing. And that's the name of the game, and that's what I want to tell you about. This is an idea that has been around for a long time, for more than 30, 40 years already. And this actually works surprisingly well. I can say that already. So the question then becomes, why is there no translation to application? Why haven't you heard about these efforts in the context of, say, cancer diagnosis or regenerative medicine? A stem cell feels different than a differentiated cell, or in the present context, the context of COVID-19, to monitor infections in blood, for example. That should all be possible, but why isn't this done? And the answer to that is that it becomes clear if you look at the techniques that have been used over the past 30, 40 years to do this kind of measurement. So probably the most simple and very robust and very good technique to measure how stiff a single cell is. Remember, cells are super small. They are about 1/10 of the diameter of one of your hairs. One of the most robust techniques is to take a pipette, a tiny pipette that's illustrated here on the left side, and you suck parts of the cell into this pipette. Right? You know the pressure that you apply, you see how far it goes in, and those two quantities can then be used to figure out how stiff or how soft that cell is. That works super well. A lot of great research has been done with that. That technique has been generally replaced by the use atomic force microscopy. It's another technique that has been actually developed to screen surfaces, topology of surfaces with atomic resolution, but it can also be abused to push into cells and see how far they can be indented with this black tip that you see underneath the yellow so-called cantilever, which bends upwards if you push down. So how far it bends upwards, that tells you how stiff the material is. That's the standard tool. I've also been involved in using something called an optical stretcher that's on the right side, that's using light in order to deform single cells. That's also quite a cool technique. The common downside to all of these, something that is shared by all of these techniques, and probably the main reason why you've never heard of doing mechanical measurements for diagnosis, is that they have really low throughput. If you measure 10 cells or 100 cells in an hour, you are really fast, right? You can imagine moving a pipette onto a cell in a microscope, sucking that in, that takes minutes, right? And then you can move on to the next cell. So it would be really difficult to get a clinical sample and screen that for abnormal mechanical cells in a clinical setting. And for that reason, I really like this quote by Sid Brenner, who said that developments in science depend on new techniques, new discoveries, and new ideas, probably in that order, meaning that you need to have the right technology before you can make new discoveries and then move new ideas forward. And that the history of science is full of that. I mean, without X-ray scattering, there would be no structure of DNA, there would be no molecular biology, right? So you need to have techniques in order to facilitate discovery and the emergence of new ideas. So this is exactly what we've done now. We have introduced a new technique that has the potential to change everything in terms of cell mechanical measurements. And this technique is actually relatively simple. It's called real-time deformability cytometry. So cytometry means that we measure cells and we measure the deformability of the cells. That's the deformability cytometry part, and the real time will become clear in a few minutes. So the setup is really simple. You have a suspension of cells. This could be blood, this could be other cells, in a syringe, and you push the plunge of the syringe out with a syringe pump typically, so that the cells flow into a microfluidic chip on a microscope. And here at the top, you see basically a top view of such a chip that contains a narrow channel through which the cells are being pushed. And here's a movie that shows you how that looks like. So you see how this cell that is initially spherical, nicely round, gets elongated as it enters that channel, and that's continues to evolve its shape until at the end it looks a little bit like a bullet. Right? So we know the forces that are acting on that cell due to the flow of the liquid surrounding the cell. So we know the forces, and we can image the deformation. And with these two things, you can then figure out how stiff or how viscous that cell is. And the technique is called real time because we can analyze the images that are coming from the camera in real time. The camera takes something like 4,000 frames a second, and we can process the images, detect whether there's a cell present, see where the contour is. This is the red line here, and that contour then gives us access to things like the size of the cell or the deformation, how strongly deformed it is. Now, I want to draw your attention to this timer that is running in the right corner there. The cell is being deformed within a few milliseconds. So that's a thousandth of a second, right? Which means that what you're seeing here is actually super slow. This is recorded and then replayed very slowly. And if we let this run at the actual speed at which this was recorded, that would look like something like this. Now, this is actually a sample situation where we analyze blood. So we've taken a drop of blood straight from a finger prick, for example, diluted in some medium 1 to 20, and then sent that through the system in the same way that I've just showed you. There's no other preparation. The cells coming in from the left side here, they widen into other channels because there's some sheath flow coming in, clear liquid that helps to center the cells in this narrow channel. And that's the channel that you just saw enlarged on the previous slide. So towards the end of that channel, we then focus our camera and check whether there's a cell in the image and analyze what its shape is. And because we're doing this in real time, we can now monitor 100 to 1,000 cells per second, right? So this is how that looks like. You're seeing a plot here emerging where on the left side, we plot the cell size, and the top, on a y-axis, we plot the deformation, how strongly deformed the cell is. And every single dot that appears here is a single cell that has been measured. There's a cell counter running here in this corner. So you see this is now 2,000 cells measured in the course of a few seconds. This is completely crazy. This is 36,000 times faster than any of the other 3 techniques that I showed you previously. And 36,000 times means if you calculate this differently, we can now measure with this technique as many cells in a single day as with the other techniques in 100 years, right? So this has changed everything that we're doing. This is a complete revolution in the field of cell mechanics. We've been able to do experiments that we never dreamed were possible. And this is just a slide to show that we have had a lot of fun publishing in all kinds of different areas with this new technique. I want to focus today on the application of looking at blood and trying to feel whether we can feel anything abnormal in the blood cells. So this is the result of a measurement of blood, similar to what I just showed you. This is, if you want, the mechanical fingerprint of blood, of a healthy donor. What do we see here? Lots of dots, so lots of cells that have been measured. In total, these are 50,000 cells that are shown in this plot here. What do we see? This group of cells here are the red blood cells. And we know that because of a number of different reasons, but one is that there are a lot of them, and red blood cells are super abundant in blood. There's about 1,000 red blood cells for every other white blood cell in blood. So this should be the red blood cells. We're also certain that they are red blood cells because they deform a lot. Red blood cells have a flat, disc-like shape, and then they're being stretched out even further. So that's why the deformation is really high of these cells. Red blood cells, you might recall, are the cells that are responsible for circulating into the capillaries of the lung, picking up oxygen, and then circulating on and dropping that elsewhere in the body, so that they are responsible for distributing oxygen throughout the body. This group of cells here down there, these are all the immune cells of the innate immune system. So these include neutrophils, monocytes, eosinophils, basophils, and a few others. The innate immune system is there basically to respond to any sort of insult to our health. If any pathogen would elicit a response of these cells, these are kind of the first responders that jump into the action right away and try to find pathogens. In the context of COVID-19, they would also be first on the scene to try to deal with the viruses that are starting to invade the endothelial cells or epithelial cells. And then there's a group of cells here that are part of the adaptive immune system. So these are the lymphocytes for the most part, T and B cells. And they are, as the term says already, adaptive. So they learn to deal with new viruses that they've never seen before, so that on a second insult, they would then know what to do exactly for that virus. So these are the cells that would recognize a virus, establish an immunity against the virus, or they also the cells that get trained by a vaccination to then respond to parts of a virus that they've been trained to identify. Okay, so this is This is essentially the mechanical fingerprint of blood according to these 3 different cell types. And what you can do easily is count how many there are. This is what you would get in a differential blood count at a doctor. When you give blood, they see, well, you have too many or not enough red blood cells. You have too many of these different white blood cells, and that is indicative of some disease. But what we can do in addition is we can check whether the cells have the right size and the right deformation, the right deformability, because that's important. This picture that you see here is very stereotypical. If you measure that between one healthy person and another, between female and male, also between relatively young and relatively old, diverges a little bit at the edges, it will always look exactly the same. And the reason for that is that these physical properties determine how the cells get around through your body. Right? So they have to have certain properties in a relatively narrow range so that they can do their job. Now, we've been interested for some time, this research has been going on for some time, in exactly checking out what we can detect, which disease conditions we can actually see. So we, for example, looked at malaria. In a patient with malaria, you see the red blood cells on the left side, this would be healthy. And on the right side with the blue cells, this is kind of the population in a malaria-infected patient. And you see that the cells tend to be less deformed than they should be. So that's a very clear signature of a malaria infection. The red blood cells are less deformed. In addition, you can also see actually the parasite in the cell directly because we're taking images of them. This is an image of a red blood cell. Apart from that, we can also detect infections. So infections of the respiratory tract, for example, we looked at that. Looked at the neutrophils, part of the innate immune system. This is a raw measurement, if you want. In black, you would see where the neutrophils would lie with a healthy donor. And in purple, you see how that population has shifted in size to become larger and deformation in response to, in this case, an acute lung injury. So if you look at the median of these 2 populations, where the center is of these 2 populations, and then plot that on the right side, you see all the gray dots. This is where the neutrophils should be in their size and deformation. And then you see how in an infection, this shifts to larger and more deformed. And if you know a little bit about statistics, these gray ellipses that you see, this is 1 and 2 standard deviations of this distribution here. So the fact that they lie far outside of that just indicates that this is a really, really strange situation that you would never find in a healthy patient. So we can use this easily to identify that there's some infection going on in a patient based on what the neutrophils are doing. The other cells, the lymphocytes, don't show much of a response. Red blood cells don't show much of a response in that case. We've also looked at things like leukemia. Leukemia is a cancer of the blood, so there are cells in the blood that are not quite mature yet, they're not fully differentiated, and they cause problems. And you can pick up these immature populations very easily. So you see this here, this would be a population that shouldn't be there. In fact, these cells should be down there in this purple circle, but they're not. But here you see the course of treatment with hydrocortisone, how the situation normalizes, and you can see how they return back to their normal physical properties. That shows that the treatment is working. This is just highlighting a few examples of what we've been doing in the past in order to show that we can really detect disease by measuring blood. Now, what I want to show you next is then how the situation looks with an acute COVID-19 infection. This was done in collaboration with clinicians at the University Hospital here in Erlangen. This was blood taken from COVID-19 patients that were unfortunately hospitalized. On the left side, you see something that I've explained already to you. This is how healthy blood looks like. On the right side, you see how the blood of a COVID-19 patient looks like. And just by looking at the 2 images, you see that there's something different, right? There are some obvious differences. I want to point out a few of them. That's what I explained earlier: red blood cells, innate immune cells, adaptive immune cells. the red blood cells should be there in this plot, but in the cancer and the disease situation in a COVID-19 patient, this population has shifted downwards, which means that the red blood cells are not as deformable anymore as they should be. Right? This is similar, reminiscent somewhat of what happens in a malaria patient with the red blood cells. This is no malaria, obviously, but the effect is the same. And the consequence of the shift in stiffness could very well be that the red blood cells don't get into these microcapillaries of the lung anymore, or not as well. If they're being pushed through the lung, they're supposed to take up blood, oxygen. So they take up oxygen. If they don't get in there, or if the surface area is reduced, they cannot take up enough oxygen. Right? This is part of the symptoms that you get when you have COVID-19. The innate immune cells should be there, and they are there, but they also have changed their properties. They are clearly showing a sign of activation of an infection. So they're larger and more deformed. That's what I showed you for respiratory tract infections previously. So that's very similar to that. There is obviously a respiratory tract infection going on. The adaptive immune cells are there. There are less of them, but there's another group of cells appearing that have clearly very different properties. In fact, they are a lot more deformed than they should be. So again, the sign that something is severely wrong. Now, we have seen all of these changes in other diseases previously, but we have never seen them in a single disease altogether. Right? This is really a massive disturbance of how the mechanical properties of the blood cells should be like. And I want to point this out. This is an acute situation, but we also looked at patients that were recalled after they had gone through the acute phase. They were recalled after several weeks and months in order to be investigated. And we saw that some of these changes, in particular what was going on with the red blood cells, persisted. So that suggested to us that maybe this is a, this is a signature of, doing this again, a signature of long COVID. Let's do this. I want to show you a couple more details here just to get an idea of how that looks like if you look at the cells themselves. Again, this is healthy. This is COVID-19 situation. This is a recovered patient that doesn't have long COVID. But you see in this diseased patient, this is how red blood cells should look like in our system. But you see a lot of cells that look very different, much less deformed, much smaller cross-sectional area. And they have problems then circulating and picking up enough oxygen. I also mentioned neutrophils, same situation. This is the normal situation. This is how the neutrophils look like in a COVID-19 patient, obviously different. When they are recovered and don't have symptoms, it goes back to normal. Monocytes are much larger. Lymphocytes are a lot softer. I mentioned that in passing already. So what that, and I said, some of these changes, especially if there's somebody that's complaining about the typical long COVID symptoms, are not going back. So what we think, what we have at our hands here is a potentially objective measure for a long COVID diagnosis. Because at present, there's no really good way of diagnosing long COVID. People complain about shortness of breath, exhaustion, fatigue, right? This is not easily quantified. These are subjective reports that they give to the doctor, and the doctor then needs to base a diagnosis on that. This might, and I say might because we still need to establish that firmly, a physical measurement that we can do on the blood of suspected long COVID patients in order to show that there is actually something wrong with something that we can measure. And in particular, this is cool because the changes that we see might actually be involved in the disease symptoms. I was kind of alluding to that already. If the red blood cells are too stiff and cannot take up enough oxygen, that would directly explain why people are fatigued, right? And if that's the case, if that is really, and I say might again, if that is really the case and we can show that, then we know what to fix. right? If it's the stiffness of the red blood cells that we need to change, then we just need to find ways to normalize the mechanical properties of the red blood cells and then hope that the symptoms go back. That's the kind of research that we're doing at the moment. So with that, I have some other piece here that I wanted to add, and that is just to motivate why we think that mechanical changes are involved in the circulation of blood cells. And that's not on blood cells, this is different cells. So this is another study that we published a few years ago where we looked at a different cell type. There are two different ways of growing them, and that difference results in very different mechanical fingerprints. So these cells, if they're grown in a three-dimensional culture, they're relatively small, but very deformed. Here, these cells, they were grown on a 2D surface, like in a tissue culture plastic, and consequently, they're a lot larger, right? These go up to 300. Here you see 300 is barely the median, and they go up to 500. But they're quite stiff. They're by far not as deformed, right? So we tested the consequence of these mechanical and size differences in a system, a macrophytic system that we tried to set up to mimic what is going on in lungs. So cells are passing through a channel with a lot of constrictions, and they are relatively similar to what the cells might encounter as they pass through the lung. Now, these are two movies, and I let them run, and I think they are rather self-explanatory. Because the cells that are small and soft, they go through this maze relatively fast, whereas the cells that are large and stiff have a much harder time making it through this maze here, through this capillary system, right? And just to put that in writing, something that you obviously would expect, that a small cell and a soft cell has a much easier time circulating through such a constriction array than a cell that is large and stiff. That's common sense. And this is a directly showing that. And we can even show that in vivo. You can inject these cells into the tailvein of a mouse, for example, and then see where these cells go. And if they are stiff, so if they're grown in this condition, most of the cells actually get stuck in the lung because it's the first organ that they go through after injection. So all of them get stuck in the lung, and hardly any of them make it to any of the other organs that in this case we wanted to target with these MSCs, such as liver, heart, spleen, or kidney. Conversely, if you take the same cells, culture them differently, and are small and soft, hardly any of them get stuck in the lung, or comparatively smaller fraction, but a lot more of them actually can then make it to heart, spleen, and kidney. So while this is not directly related to COVID-19, I think it very nicely illustrates this idea that the mechanical properties of the cells, the size, the stiffness, etc., is important in considering where cells can go in your body, in particular red blood cells through the lung and then to other organs. But the same is also true for the immune cells, obviously. So with that, I come to the end of my short presentation here. I think the take-home messages could be that this approach to do a physical measurement, a mechanical measurement on cells is both a sensitive and also a marker-free way to characterize cells and their function. In particular, this technique that we've introduced, real-time deformability, is a high-throughput way of measuring these physical properties that we think has the potential to revolutionize cell-based diagnostics. And in the context of the present situation, COVID-19 and also long COVID seems to have a very specific and peculiar blood mechanical signature that could be used for diagnosis. We're currently involved in several studies in order to establish that firmly, because obviously if you only measure, say, 20 patients, that might not tell you what is going on in a population of a few 100,000 people that have that condition. And so we need to screen a much larger cohort of these patients, which we're doing at the moment. And that also opens the door to trying to identify sensible therapeutic approaches to reverse the changes in the blood cells, and then hopefully also to return to normal in the context of the symptoms that these patients are suffering from. So that's what we're doing at the moment. I want to very briefly also show my team. We are on Twitter if you want to follow us. And that's what I wanted to share with you today. Thank you very much.
Thank you very much, Jochen. Excellent. I thank you so much for also give us the proper background in understanding the measurements. I think it's very impressive the way you show the link between physical properties and physiology, which is not so easy to see for me sometimes in just in the biochemistry. But I mean, you even see the mechanochemical coupling. You see the effect of was it in the leukemia patient, how the mechanical properties recover with treatment. Correct? Did I understand it correct?
Yes, exactly.
Yeah, I think this is really impressive. Although I think you explained almost everything perfectly, I still have a few questions, if I may. Of course, of my interest of how one interest is of how you planning to continue. Are you like effects of like have a correlation between like let's say the antibodies or the vaccination state of a patient with the mechanical properties? Are studies like that are planned or something like that?
Vaccinations, we haven't done. That's an obvious one. I think our main problem is that there are too many things that could be done, and that would be really cool to investigate. And there are only so many people involved in this research at the moment, and only so many instruments available. So one major push at the moment is to try to simplify the measurement device to the point where this could be given into the hands of a lot of other researchers, so that this could be put on a much broader science base. But until that's in place, it will take certainly, I don't know, half a year or something like that. We are collaborating with a lot of people in Erlangen and elsewhere in Germany, also elsewhere in the world, to try to just increase the data situation and database. Right, we have a cohort here in Erlangen where we are measuring 5 to 8 COVID-19, the long COVID patients a day. Just, you know, that will ramp up the numbers relatively quickly, and we'll have statistical power to say something very definite about this. We are also starting to be involved in certain therapeutic interventions for long COVID. You mentioned autoantibodies, and that's a very interesting one. Colleagues of us here in Erlangen, Bettina Hoberger in particular in the eye clinic, she's been doing a treatment study using a certain experimental drug to inhibit the activity of autoantibodies that are directed against G protein coupled receptors. And the first treatment results seem to be rather encouraging, where the symptoms have reversed to normal within a few hours and persist on a single dose. She's an eye doctor, and that means she also looks at things like the perfusion in the retina using OCT and angiography. So she sees that the perfusion that is disturbed in both COVID-19 and also long COVID patients normalizes with the treatment. And we now have started to measure the first, these are 2 patients treated, so it's really, really low numbers. So everything to a grain of salt or probably a sack full of salt. Also the mechanical properties normalize.
Okay.
So that's very encouraging. And obviously there's a lot that still needs to be figured out in terms of mechanism, whether the autoantibodies act on the cells directly, if they act on the endothelium, and there are some other humoral factors or whatever, that then also control the cell mechanical properties. Obviously that's a correlation that we're starting to establish, but the causation is a much more difficult task to show that it's really the stiffness of the cells that prevents them from getting somewhere. But I mean, everything looks promising so far, and we're in a position, we have the tools, we have the right people together to try to investigate that. So that's what we're doing at the moment.
I mean, the macroscopic link is impressive. I mean, that you have, but maybe along those lines, my question, another question is, do you have any studies going on or collaborations where you look into what part changes in the cell that changes the deformation, like the cytoskeleton or some parts of that? And so, so also interested, would other part, would for example the endothelial cells or the entire vascular system also change?
That would be the initial hypothesis, I think, for most of the doctors that are involved. When they say circulation is different, perfusion is different, or the endothelial surface is different, it gets more sticky, the the vasculature contracts or collapses, and that's why there's no blood cells going through. So what we are offering is an alternative orthogonal aspect, and I suspect both of them will play a role, right? In terms of what is going on in the cells that changes them, in the white blood cells, it's certainly the a combination of the actin cytoskeleton that is involved in the activation of the cells. And I think the signaling pathways are relatively clear. We have not looked specifically at the situation, what's going on, but I think that is an obvious thing to follow up on. It could have to do with ion regulation, ion pumps, etc., as to the extent that they exist, considering your other research. And so, but there's some osmotic regulation of the size of cells that's also involved. And for the red blood cells, I think that's mostly lipid composition. So the composition of the phospholipid bilayer that will be involved in the deformability or changes in the deformability. So any sort of research looking into lipid composition in cells, in the red blood cells, might be an interesting aspect to look at in terms of figuring out what is actually going on with the cells.
Excellent. Now that's something a lot of people in my community probably love to study and hear. Yeah, I think this was impressive. Also, you were mentioning that you have you see changes in all the cell types you're looking at. That this is quite unusual from your perspective. And indeed, in this this paper I was citing at the beginning of my little introduction is that that you see in all kinds of organs, in all kinds of on all kinds of levels, you do see impact. So this maybe this is where the story also is linked together. I want to thank you here, Jochen, for this very exciting story, and please keep me up to speed, or keep us up to speed. For all those listening out there, stay tuned. The next episode will come up sometime within the next couple of weeks, and this will be on the latest news on aerosols and how this is related to COVID-19. Jochen, thank you very much. Was great to have you. Bye-bye.
Bye.